Hello students, today we are going to read about the Lac-Operon model, then human genome project, and then after DNA fingerprinting. Okay, before start to this Lac-Operon model, first I want to tell you about the genes, right? And what is here you can understand here means because lac means here lag means what? Lactose sugar. So this operon means it is a small fragment of DNA in which one or more than one structural gene is there, one promoter, one regulator, and one operator gene is present, right? So this one is called a operon. That segment, that DNA segment is called operon, right? In which at least one or more than one structural gene, one promoter gene, one regulator gene, and one operator gene is present. Okay, but first, before going to gene, what is gene and how many types of genes are there? First, we understood this one. When we consider gene in our body or in any cell, not our body means any organisms, we actually it has two types of genes, right? One is called constitutive genes, and another one is called non-constitutive genes. You can understand here. Here, constitutive genes means what? These are also called as housekeeping gene, right? And non-constitutive genes means they are also called as smart gene or deluxe types of genes. On the basis of their function, right? On the basis of their function, we have two types of genes. We can say two types of genes. Which one? First one is, I tell you, constitutive, right? It is called as constitutive gene. Constitutive genes, they are also called as housekeeping gene. Housekeeping, right? Housekeeping means this. Look here, our domestic works are compulsory works. So they are continuously going on day by day, every day, right? And here also, this constitutive genes, it is constantly going on. Means these genes are active, constantly, and they are essential for their metabolism, and are, we can write here, and are essential for metabolic activities. Metabolic activity is essential means that for, as for example here, like our and that is means we can say gene of glycolysis. Glycolysis means we know in glycolysis different type of enzymes are required. So these enzymes, they are that means they are produced by specific type of DNA, and these DNA they are continuously working. So they work continuously, no stop is there, right? Or we can say ATP synthase, genes of ATP synthase, because ATP synthase means it is also continuously formed in our body, in our cell. So these genes, they are continuously working on, they have no switch on, switch off is there, they never rest, right? So these are called as housekeeping gene or constitutive genes. And then the second gene is our non-constitutive genes. Non-constitutive genes, they are also called as smart gene or luxury genes. Smart genes or luxury genes, right? Smart gene or luxury gene, why they are called as non-constitutive? They are two types. These are two types. Two types on the basis of their function, right? One gene, one is called as inducible gene, and second one is called repressible gene. Inducible gene and repressible gene. This inducible gene, this one, right, and repressible gene, these both. What inducible means? This one, right? It is mostly, oftenly it is non-active, that is inactive. This commonly inactive, right? But in the presence of inducer, or when the cell demands, right, means it is, these are actually inactive genes, but when the cell demands, or when we can say when inducer is there, right, so due to the presence of inducer, they become activated, right? So they are commonly off position, means commonly inactive, means we can say it is switch off, commonly switch off. But due to the presence of inducer, or when we can also say when cell demands, then what happens? It activates. Then it activated. So this one is called inducible, right? Means at that time, activate means what? It come into the on process. And just different is repressible. Repressible means what? They are commonly active, normally active, means they are normally always in switch off process, switch on process. This one is switch off, right? Means normally it is inactivated, but it is normally activated. But when the substrate becomes more evident, means it forms sufficient amount of substrate is formed, then what happens? The excess amount of substrate repressed or inhibit the genes, right? Or we can say in the presence of co-repressor. In the presence of co-repressor, this one is stopped, means it switch offs, right? So normally it is inactive phase, but due to the presence of repressor, it inhibits the production or it stops, right? Means it becomes inactivated, right? We can write here due to the presence of suppressor or repressor we can use, right? Repressor. Mostly these repressors are substrate, right? Or we can say end product. That means it is also we can say end product, right? This process is switched off. This process is switched off. SWITCH, switched off, right? This one is switch on, but this one is switch off. So we have two types of genes, you understand, constitutive genes or housekeeping genes. They are constantly work, never stops, right? And another gene is our non-constitutive genes, or we can say smart genes, or we already tell them luxury genes. So it is also two types. One is continuously work, another one is not work. Continuously work means what happens when the cell demands to stop this working, then what happens? These genes, they stop their functioning, right? Means these are continuously in switch on process, but when substrate is formed more than sufficient amount, then what happens? This substrate, it acts as repressor or co-repressor and stop this process. As for example here, this repressor gene example is tryptophan operon, right? And as for example here, another one is our inducible. Inducible means what? Inducible means they are normally inactivated, but what happens in the presence of inducer or when cell demands, then what happens? It become activate, means it switch on process, and what happens? It start the formation of proteins or polypeptides, right? Here, this one is, as for example, lycopyran. So these are the two examples of two different type of non-constitutive genes. Do we understand? Okay. Now come to the lycopyran model. Means in our NCERT lycopyran model, NCERT line by line. The elucidate lycopyran model means, okay, here operon. This operon word is used. This term is used by Jacob and Monod, right? These two are Francis. That means these are French microbiologists. Is given here. They are geneticists. Is given. The elucidation of lycopyran. This operon term, lycopyran term or operon term is given by this Francois Jacob and a biochemist, that is Jacob Monod, right? Simply we can say Jacob Monod was also a result of close association between the geneticist. Who is geneticist? This is Francois Jacob, and a biochemist. This biochemist is Jacob Monod, right? They are French, right? Means scientist. We can say French scientist, and they are also working at the same university. Same university means which university? It is Pasteur University in Paris, right? They were the first to elucidate the transcriptionally regulated system in lycopyran. Here, lac refers to lactose. Lac for lactose sugar, right? Here, trip means what? Tryptophan sugar. A polycystonic structural gene is regulated by a common promoter and regular gene. Look here, a polycystonic structural gene, because this one E. coli, this lycopyran model is present in E. coli, and E. coli means what? It is Escherichia coli. It is a bacteria, and we know bacteria or prokaryotic cells, they have which type of messenger RNA? They have polycystonic messenger RNA, right? Cystone means what? Cystone means functional unit. If more than one functionality is there, then we can say it is polycystonic. And eukaryotic means we have monocystonic, right? Here what is given? Polycystonic structural gene. Polycystonic structural genes means what? It contains 3 genes. Which genes? It is X, Y, it is Z, Y and A. This polycystonic gene, right? This polycystonic gene, it contains Z, Y, A. 3 genes. Each one is called cystone. We can say here 3 cystones are there. One is Z, one is Y, one is A. That's why it is called as polycystonic structural gene, because it contains more than one gene. Structural gene, right? Such arrangement, and also other genes are also present, that is regulated by common promoter and regulatory genes are also there, and operator gene is also there. Such arrangement is very common in bacteria, and it is referred as to operon. Now we understand operon means which genes are present in operon? A structural gene which contains one or more than one cystone, right? Second means what? Promoter gene, one single promoter gene, single regulator gene, and single operator gene. To name few such examples are lycopyran, lactoseoperon, tryptophan. I already tell you this is the repressor example of repressible operon model. Tryptophan operon, araoperon, his means what? Histidine operon, val means I think valine, right? Operon, extra, right? The lycopyran consists of one regulatory gene. That gene is I, right? One regulatory gene, that is I gene. Here, the term I does not refers to inducer. It is not a inducer because it produces a inhibitor, repressor protein. That's why here it is not inducer. Rather, it is derived from the word inhibitor, because it produces a messenger RNA, and this messenger RNA produces a repressor protein, right? And this protein is responsible for switch of the process, right? To stop the process, to stop the transcription. That's why it is called as inhibitor. Okay. And the 3 structural genes, that 3 cell means Z, Y, A. I'll tell you that is Z, Y, A, right? The I gene code for the repressor, and the 3 structural genes, that is Z, Y, A, Here simply it is given this small I, it is our inhibitor gene and it produced repressor, right? And our three genes, that is Z. Z means what? It produce Z. Here, look here. Always we go from Z, then Y, then A. What is, when you consider the tryptophan operon, also it is reverse order. That is reverse Z Y. It is not A Y Z. It is Z Y A. When we consider tryptophan operon, it is also reverse order. E D C B A, not A B C D E. It is E D C B A. Right here, Z means what? It is not A2. It is Z means what? Beta galactosidase. Right? Beta galactosidase. Beta galactosidase. What is the function of this beta galactosidase? It digests the lactose sugar into, because lactose sugar is a disaccharide. It is made up of one glucose molecule and one lactose molecule. So this beta galactosidase enzyme, it breaks the lactose sugar into galactose and lactose. Okay. Second is our Y. It is responsible for the formation of a enzyme, that is permease enzyme. Name, according to name you understand permeability. It is responsible for permeability, means it make permeable, that is lactose sugar is it increase the permeability of lactose sugar, because lactose sugar is present outside the cell, right? So this lactose enters into the cell. So how it enters? Due to this enzyme, that is Y. It is called as permease, right? Permease enzyme means it is responsible for the transportation of, or it increases the permeability of cell membrane for lactose, right? Z gene code for beta galactosidase, it is given here. BGAL, beta galactosidase, simply we said them BGAL, shortcut, which is primarily responsible for the hydrolysis of disaccharide. Disaccharide means which one? Lactose. Already I tell you in the biomolecules that disaccharide means how many disaccharides? Period here, lactose, maltose, right, and the sucrose, right, and that is, we can say these are, right, disaccharides. Here lactose is there. Lactose means milk sugar into its monomer unit, that is galactose and glucose, right? The Y gene code for permease enzyme, I write here permease enzyme. Why it is? Which increase the permeability of the cell to B galactosidase, right? It increase the permeability of the cell to B galactosidase, right? So B galactosidase, it is responsible for the catabolic process, means it breaks the, or it digests the lactose sugar, but first it has to transfer from the cell wall. Okay, the A gene, that is the last gene. Okay, this one A means acetyl. Here the A gene encode the transacetylase. What is the function of this transacetylase? Here it is not given, but I tell you, it forms transacetylase. So transacetylase, what is the function of this transacetylase? Trans means transfer. It is responsible for the transfer of acetyl group, right, from acetyl CoA enzyme. Acetyl CoA enzyme, right? Acetyl CoA enzyme. It is responsible for the transfer of acetyl group from acetyl CoA enzyme. Hence, all the three gene products in lacoperon are required for metabolism of lactose. Metabolism of lactose means for the digestion of lactose sugar, right? In most of other operons as well, the genes present in the operon are needed together to functions. They function simultaneously and coordinately, right? To function in the same or related metabolic pathway, and the figure is given. In this figure you can understand how a lacoperon works, right? Now look here. Here you can see this one is a DNA. It is our DNA, not our means E. coli DNA, right? It is the DNA of E. coli, right? You can see here P for promoter gene. This one is promoter gene. Promoter gene. What is the function of this promoter gene? Promoter gene, this site, this is the promoter site, we can say. So this is the promoter site where RNA polymerase enzyme binds. RNA polymerase enzyme binds at this site, right? Now this one is regulator gene. Regulator gene. Regulator gene, this I already it is given that I is not for inducer, it is for inhibitor. It forms, because RNA polymerase from this DNA, it forms a RNA. This is the repressor mRNA. Messenger RNA is formed here. So this process is called transcription. Right? So through transcription, a repressor mRNA is formed. Now this mRNA, it forms repressor protein. This one is repressor, it is a specific protein. And we know this process is called translation. So through translation, repressor mRNA forms a repressor protein. Now repressor protein, in the absence of any inducer, what happens? This repressor protein, it binds with operator gene. This one is our operator gene. Right? In the absence of any inducer, this repressor protein, it binds with operator. When it binds with operator, then what happens? Switch off. This process is called switch off. Now look here, RNA polymerase, which move from this to this side. Then what happens? Due to the presence of this repressor protein and operator gene, right? It cannot move or it cannot pass the operator gene, and that means what? Transcription of ZYA is stopped. That what happens here? No transcription. That is no transcription. This, right? Because why? This process called switched off, right? Repressor binds with operator. Normally these genes is stop, is present in the stop condition. No transcription happens here. But if lactose sugar is there in the environment, then what happens? Look here, lactose sugar, for switch on of this process, lactose is present every time in the cell. Okay, it is very little amount it is present, but it should be present. It must be present inside the cell. Otherwise, this operand model never starts. Okay. Now here, in what happens? When an inducer is present, then what happens? This promoter gene, RNA polymerase bind to this site, right? Then what happens? It moves to this side. When it moves to this side, repressor mRNA is formed through transcription. Repressor mRNA is formed, right? And this repressor RNA, it forms repressor protein. This one is protein. Repressor protein, right? It forms through which process? Translation process. This is transcription. This one is translation process, right? Now what happens? Because inducer is already present here. So this repressor protein, because it is a three-dimensional structure is there, right? And this protein, it has active sites. This inducer, it binds with active sites of repressor. Now this is the inducer. That means we can say it is lactose, right? And this lactose, it binds with repressor protein and forms a inducer repressor complex, right? Now it become inactivate, because here all the active sites of this protein is totally filled up with lactose sugar. Then it is unable to bind with operator gene. It is unable to bind with operator gene, and this one is operator gene, which is free. Operator gene, free operator gene, right? Because here no repressor protein bind with this one. Now what happens here? When polymerase enzyme starts, then what happens? This promoter site, right? This is another promoter site. It is here. It is the site where RNA polymerase binds with promoter. This one, RNA polymerase binds with promoter site, right? RNA polymerase, when binds with promoter site, it starts transcription. Now this transcription, it passes over the operator and transcript the ZYA gene continuously. Now Z specific part, this mRNA, this is the lag mRNA and this is the transcription, and this mRNA is polycistronic mRNA. Polycistronic mRNA because it contains three different genes, three different cistrons. Three different cistron means ZYA, and through these three different genes, three different type of proteins are formed, or we can say polypeptide chains are formed, or we can say enzymes are formed. That from this gene, which one is formed? Beta galactosidase is formed. From Y, permease is formed, and from A, transacetylase is formed. This process is called translation. Three different proteins are formed. Here what happens? Three different polypeptide, or we can say enzymes are formed. Enzymes are formed because it contains three genes, three cistron bodies, that was three cistron units. So we can say this is a polycistronic messenger RNA. Do you understand here? Right? This process, or we can say this one is this operator, this operon model. It is a inducible model. This gene is example of inducible gene. Right? Why I said here it is a inducible gene? Because due to the presence of inducer, right, it starts transcription. Right? Do you understand? Normally it is in inactivate form, but when cell demands, when cell needs these enzymes, which one? Beta galactosidase, and this enzyme is, what is the function of this enzyme? It breaks. When cell needs the digestion of lactose sugar, when the cell has no glucose, then it has to digest the lactose sugar, then it produces the beta galactosidase enzyme. For the production of this beta galactosidase enzyme, it needs one lactose sugar at least, right, which act as inducer. Right? Do you understand? Then it starts the formation of enzymes. Right? And this means we can say this is activated, this lag operon model, it is activated until the lactose sugar is present inside the cell. It can continue going on, right? Or the cell demand it. So do you understand this operon model? It is very easy. Lactose is substrate for the enzyme beta galactosidase. Lactose sugar is substrate because it is why it is substrate? Because it has to digest, right? It is broken down into. So lactose is the substrate for enzyme beta galactosidase, and it regulates switching on and off of the operon. When lactose binds with repressor, switch on, and when lactose is not bind with repressor, this one is switch off process of the operon. Hence, it is termed as inducer. When it binds in the presence of lactose sugar, this transcription starts. So it is called switch on, or we can say inducer. Right? In the absence of preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, no glucose is there, no lactose is there, then what happens? This bacteria first it has to digest the lactose into glucose and galactose, right? So the lactose is transported into the cell through the action of permease. Now you understand what is the function of permease? The lactose is transported into the cell through the action of permease. Means permease increase the permeability of cell membrane for lactose and also for beta galactosidase, right? Remember, a very low level of expression of lac operon has to be present in the cell all the time, otherwise lactose cannot enter the cell. To start the lac operon model, lactose is present all the time in the cell, right? The lactose then induces the operon in the following manner. The repressor of the operon is synthesized. Repressor, it is continuously synthesized. So repressor of the operon is synthesized all the time, and that is called constitutively. Now you understand? I tell you, constitutive genes. That means here we can say this I gene is an example of constitutive genes because it is continuously forms all the time, continuously forms the repressor gene. right? Repressor protein. Messenger RNA, it forms the repressor messenger RNA, repressor messenger RNA forms the repressor protein, right? This protein is continuously formed and this protein is continuously binds with operator gene. So, what happens? Lac operon is switched off. Means it is normally it is in switch off position. The repressor protein bind to the operator region of the operon and prevent the RNA polymerase from transcribing the operon, right? In the presence of an inducer such as lactose or allolactose. Actually, allolactose is the real inducer, right? But in the absence of allolactose, lactose is there, they are isomers. The repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeded, right? Essentially, regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate, right? Here, regulation of lac operon can also be visualized as the regulation of enzyme synthesis by its substrate, because substrate, lactose is a substrate to be digested, right? And it is also regulate the lac operon. Remember, glucose and galactose cannot act as inducer. That is important. Glucose and galactose cannot act as inducer for lac operon. Only lactose or allolactose is the inducers, right? And chief means when we consider which one is the main means allolactose, right? Now, and the other, can you think for how long the lac operon would be expressed in the presence of lactose? It is depend upon the availability of lactose. First thing is there. Second, and the depend upon the demand of the cell. When a sufficient amount of glucose is formed inside the cell, then it is stop the process, right? Second, when lactose is absent, then also due to the absence of lactose cannot bind with repressor protein. That means what? It is stopped. Repressor protein bind with the operator and this process is stopped. Okay. So, regulation of lac operon by repressor is referred to as a negative regulation. Regulation of lac operon by repressor, right, is referred to as negative regulation because it is switched off the process, right? Lac operon is under control of positive regulation also. Lac operon is under control of positive regulation as well, but it is beyond the scope of discussion at this level. You can read, you will be read in your next classes, higher classes, right? So that means what? It is not for negative only. It has also some function for positive. Basically, it is negative because due to the presence of this repressor, this means that is formation of messenger RNA, or we can say transcription stops. That is switch off process. So you can say it is a negative, but it is also controlled in the positive regulation as well, right? Positive regulation means when lactose is present. That what happens? Lactose, it is a inducer. Inducer means positive. Because already it is switch off, then it start. Start means one positive is there. Positive and negative both are present here. But chiefly it is negative, right? Negative regulation. Negative means because this negative means what? Due to the presence of repressor protein, due to the absence of lac operon. Negative effect means what? It is stop the process, right? And if lactose is present, then positive feedback is there. Positive means what happens? Lac operon stops, right? Do you understand? Another example is that is one example for lac operon model. Another example is as for tryptophan operon. So tryptophan operon means what happens here? In this case, means in lac operon model, what we read here? In lac operon model means what? Here we can say it is repressor protein, repressor plus inducer, right? Then what happens? Switch on process. It is called switch on, right? And when we consider the tryptophan, this is the lac operon. That means we can say inducible type of operon, right? But in case of repressible, what happens here? Here, aporepressor plus corepressor. is equal to what happens? Switch where on, but here what happens? Off process is there. Switch off. They become active. Apo-repressor plus co-repressor, they becomes active apo-co-repressor complex. Here what happens? Repressor plus inducer, what happens? Inducer, repressor, inactive inducer, repressor complex is formed, right? Now next point is our Human Genome Project, HGP. In the preceding section you have learned that it is the sequence of bases of DNA. We know DNA means what is the sequence of bases, right? That determines the genetic information of a given organism. In other words, genetic makeup of organisms or an individual lies in the DNA sequence. Totally it is present inside the DNA sequence. If two individuals differ, then their DNA sequence should also be different, right? At least at some places, not whole, but some places, right? These assumptions led to the quest of finding out the complete DNA sequence of human genome with the establishment of genetic engineering techniques, and where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining the DNA sequence, right? A very ambitious project of sequencing of human genome was launched in the year 1990. In which year? 1919, HGP Human Genome Project starts. It is launched, right? Human Genome Project was called a mega project. It is a mega project. You can imagine the magnitude and the requirements for the project if we simply define the aims, right, of the project as follows. What is given in NCERT? Look, here only statement type question will come, right, from this chapter, from this point, right. Here you understand, human genome is said to have approximately, we know very well, 3 into 10 power 9 base pairs. This 3.3 into 10 power 9 base pair in case of haploid. But and if cost of sequencing requirement is US $3 per base pair, on the $3 per base pair, the estimate cost in the beginning at that time, right? The total estimated cost of the project would be approximately $9 billion US dollar. How much? $9 billion US dollar at the time, at 1990, right? Further, if the obtained sequence were to be stored in typed form in the book, then we need each page of the book contains 1000 letters and each book contained 1000 pages. Then we need 3300 such books would be required to store the information of DNA sequence from a single human cell, right? The enormous amount of data expected to be generated also necessitate the use of high-speed computational devices. Computation means computer, for data storage and retrieval and analysis. Human Genome Project was closely associated with the rapid development of a new area in the biology that is called bioinformatics. It's called bioinformatics, right? Now goals, what are the aims or what are the goals of HGP? Some of the important goals. How many goals are there? Here 6 goals are here. What? Identify all approximately 20,000 to 25,000 genes in human DNA. At that time, we have no any idea how many genes are there, right? Because I know when I read in 1992-93, that time I read that 1,20,000 genes are available in human body. But after the Human Gene Project that is completed in 2006, okay, this time is only, it is a 13-year project, right? But it is completed by 2006. Last 1990, completed 2003. But actually, practically, right, it is completed in 2006 May. Okay. So after then we can then we realize that it is not 1,20,000, but it is only 30,000 genes are there, right? Identify all approximately 20,000 to 25,000 genes in human body. You have to read this, right? 20,000 to 25,000. It is given in human. Look here, in other, when we read the human genome project after, then there it is given 30,000 genes. Here 25,000. But we have to read the stack statement of NCERT, because the questions which come into the NEET paper, it comes from exact. That means they need only exact line of NCERT, right? This is the only prescribed book for us. So determine the sequence of 3 billion chemical bases pairs make up human DNA. Look here, 3 billion. It is not a million, it is billion, right? Third, store this information in database. Fourth one is improve tools for data analysis. Fifth, transfer related technologies to other sectors such as industries. And last one, because some social issues come, then what happens? Address the ethical, legal and social issues. Ethical, legal, social issues, simplify in a simple word, or in short form we said them LSI, ELSI, E for ethical, L for legal, S for social, I for issue that may arise from the project. How many goals are given here? 6 goals. First, identify 20 to 25,000 genes approximately. Second one is determine the sequence of base pair. How many base pairs is given here? 3 billion base pair, right? Third one is store this information into a database. Fourth one, improve the tool for analysis. Fifth one, transfer related technologies, right? And sixth one is address the ethical, legal, social issues, right? The Human Genome Project was a 13-year project. That means what? It launched in 1990. It would be completed in 2003. But it is not completed in 2003. It is completed in 2006 May, right? And last, the human gene was last chromosome which is detected. It is chromosome first, which one is the largest one also. Coordinated by the U.S. Department of Energy and National Institute of Health. U.S. Department of Energy and National Institute of Health, right? Then what happened? During the early years of the HGP, Human Genome Project, the Wellcome Trust, very good name is there, Wellcome Trust from United Kingdom, right? It comes, become a partner, become a major partner. Then after what happens, slowly the other countries will also come, right, and becomes a partner. As for example, additional contribution came from Japan, France, Germany, China and others also. Japan, France, Germany, China and others, they also contribute. Means here we understand US Department means United States, that is our America. Department of Energy and National Institute of Health. Then who came? Then Wellcome Trust come from UK, United Kingdom. Then after Japan, France, right, another Germany and this China, they also came. The project was completed in 2003 because it is a 13-year project, so it is completed in 2003. Actually, it is not completed in 2003, right? On paper, it is completed in 2003 because it is a 13-year project. Actually, it is completed in 2006. Okay. Knowledge about the effect of DNA variation among the individuals can lead the revolutionary new ways to diagnose, treat and someday prevent the thousands of the genetic disorders or genetic disease, we can say here, right? That is disorders, that is genetic disorders or genetic disease that affect the human beings. Beside providing clues to understanding human biology, learning about non-human organisms, DNA sequence can lead to understanding their natural capabilities that can applied toward the solving challenges in health care, in agriculture, energy production, environmental remediation, right? So, a non-human genome project is also established. Many non-human model organisms as for some examples given here, such as bacteria. Which bacteria? E. coli, yeast, scientific name is Saccharomyces cerevisiae. We can say the first prokaryotic cell which is come in genome human, that is genome project DNA sequences, it is called E. coli, right? Second, first eukaryotic cell, it is yeast, right? Another is animal. First animal is Caenorhabditis elegans, which is a free living nematode, non-pathogenic. Free living means non-pathogenic, already it is, right? Nematodes. First animal, first eukaryotic cell, first prokaryotic cells, and first, that is our another animal, that is Drosophila, the fruit fly, Drosophila melanogaster, right? Plant means rice and Arabidopsis. So Arabidopsis, rice, very well, rice. In earlier, what happens? In earlier, this rice genome project is also present in our syllabus, but this time it is deleted, right? Arabidopsis means it is the scientific name of small mustard. Small mustard, right? Small mustard type, right? It is Arabidopsis. Extra, have also means sequenced. So, do you understand here what is given? The first bacteria, it is E. coli, that is sequenced. First bacteria means first prokaryotic cell. First eukaryotic cell means yeast, Saccharomyces cerevisiae, sequenced. First animal is Caenorhabditis elegans. This is a free living nematode, non-pathogenic. Another one is Drosophila, right? Plants, first plant is Arabidopsis, not rice. First plant is Arabidopsis, that is small mustard plant. A small variety of mustard plant is there, and then after rice is there, right? Have also been sequenced. Okay. Now methodologies. What is the method for this method which is used for here, right? So methodologies. What is methodology here? The methods. The method involved two major approaches. Two major approaches means first we sequenced the only expressed. Expressed means which can express, which is responsible for the phenotypically changes or anatomic differences, which are transcriptionally active. Simply we say, which are transcriptionally active, we can only sequence them. Second means what? We can sequence all the, means either it is transcriptionally active or inactive, that is not a matter. We have to find out the sequence of all the nucleotides, all the base pairs. That is here two. Simple. The method involved two major approaches. First, one approach focused on identifying all the genes that are expressed as RNA. That are expressed as RNA means what? They transcript RNA, or we can say they are transcriptionally active DNA, referred to as expressed sequence text. ESTs. ESTs, expressed sequence text. Express means which can express itself. The part of DNA which can express itself in the form of messenger RNA, or we can say which is transcriptionally active. The other took a blind approach of simply sequencing the whole set of genome that contains all the coding and non-coding sequence, sequences, and later assigning the different region in the sequence with functions, a term referred to as sequence annotation. So, two major approaches are there. First, expressed sequence tag, only genes which can express in the form of messenger RNA. Second, all the region of DNA means all the nucleotides which are present in DNA will be sequenced. For sequencing, the total DNA from a cell is isolated. First means what? First, we have to isolate the DNA, right? So we isolate the DNA due to the help of different digestive enzymes, right? RNase enzymes, protease enzymes, ligase enzymes, right? And then centrifugation, and convert into random fragment relatively smaller size, right? Convert into random fragment relatively smaller size by the using of restriction endonuclease enzymes, right? Recall DNA is very long polymer and there are technical limitations because we have no any technical method to sequence the long polymer. right? Sequencing very long pieces of DNA, we have a limitation, right? And cloned in a suitable host using specialized vector. We cannot clone the whole DNA. Only we can clone a small part of DNA. Okay. The cloning resulted into amplification of each pieces of DNA fragment so that it subsequently could be sequenced with each. The commonly used host were bacteria and yeast, right? And that's why we give them name. The vector we are called as, which are inside the bacteria, they are called as bacterial artificial chromosome, or yak means yeast artificial chromosomes. Do you understand this one? Now next passage, it is given the fragment were sequenced using automated DNA sequence that work on the principle of the method which is developed by Sanger. Its full name is Frederick Sanger. And this Sanger, it is also credited for developing the method for determination of amino acid sequence. And he get, look, this one. Fragment were sequenced using automated DNA sequence that worked on the principle of a method which is developed by Frederick Sanger, right? This Sanger is also credited for the developing method for determination of amino acid sequence in the proteins. Amino acid sequence in the proteins. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments. For sequencing, alignment of these sequences was humanly not possible. We cannot do this because they are very small part. How could we recognize, right? First thing. Second, first very small. Second, what is there? Because our sense organs are not able to differentiate or to recognize these sequences, right? Therefore, a specialized computer-based program were developed. These sequences were subsequently annotated and were assigned to each chromosome, right? The sequence of chromosome first was completed only in May 2006. This sequence of chromosome first was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes, 1 X which is in female and 1 Y. 22, 23 and 24 human chromosome sequence where it is completed in May 2006 as I tell you. Now you can see here one cell from this, a girl is here in the figure, right? One cell. In one cell what happens? We isolate the DNA. First process means here cell, then what happens? Isolated DNA. We can say here isolation of DNA. When we get DNA, look here, this DNA. Now here this have, what is this have? Nitrogen base pairs. We can say base pair, right? And this is this we use computer base pair. What is given? TG, TA, CG, TA, CA, right? A representative diagram of human genome project. How could we understand this? By the using of computer. Without computer, it is not possible. Okay. Another challenging task was assigning the genetic and physical map of the genome. Assigning the genetic and physical map on the genome. Genome is what? This was generated using information on polymorphism of restriction endonuclease recognition site. Polymorphism means many forms, right? of restriction endonuclease recognition sites. A specific restriction endonuclease cut the DNA to a specific sequence, right? We can say specific region. Specific region means why it is called specific region, because it has a specific sequence. Either it is made up of 4 base pair sequence or it is made up of 6 base pair sequence. E. coli, that for E. coli, it cut the DNA where 6 palindromic sequence is there. But when we see the He for HAE3, right? The He3, it is mostly used in DNA fingerprinting, human DNA, means human, right? As a human restriction enzyme. What happens here? He comes from Haemophilus aegyptus, right? 3. It cut the DNA where 4 palindromic sequence is there. It is GGCC is there. And in DNA fingerprinting, commonly which is used, is most common restriction enzyme which is used, it is HAE3, right? Haemophilus aegyptus 3, okay? And some repetitive DNA sequence known as microsatellites. One of the application of polymorphism in repetitive DNA. Repetitive means where the base pair, we can say here, repetitive DNA sequence means what? It is the stretch of DNA, it is a part of fragments of DNA which repeats many times. In this repeat DNA, there is a sequence, specific sequence is there, right? Then this sequence, nitrogen base sequence, it is made up of 6 to, we can say 11 to 60 base pairs, right? So they repeat many times. So they form a that is length of this repeated DNA. It depends upon the we can say here VNTR, variable number of tender repeats. How many times they repeat? That is depend upon the this how many times means that number of VNTR is responsible for the means we can say this restricted fragment length. Okay. Also shall be explained in the next section of DNA fingerprint. We will read this one, right? Silent feature of human genome. So what feature? After this human genome project, we get some features of human genome. What is given here? You can we have to read here. How many given? Here 9, right? 9 features are given. So we read this. The human genome contains 3164.7 million base pairs. First, very important, 3164.7 million base pairs are there, right? It is million, not a billion. Okay, 3 billion, but 3164 exact number. Second, the average gene consists 3000 bases. A single gene, average gene is not. It consists 3000 bases, but size vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. Dystrophin, right? It is also called as Duchenne muscular dystrophy, right? DMD, Duchenne muscular dystrophy. The largest gene, right? The total number of gene is estimated at 30,000. Second, what is given? Total number of estimated gene, it is how much? 30,000. This time, after human genome project, we realized only 30,000 there, much lower than previous estimates of 80,000 to 140 genes. I already tell you when I am in the intermediate classes at that time, like you. At that time, I read a human gene means it can, how many genes are there? It is near about approximately 1,20,000. And we understand here what is given here, 80 to 140, average means what? 1,20,000 genes are there. But this time, after human genome project, we realize or we understand that only 30,000 genes are there. Almost all 99.9% genes or nucleotide bases are exactly same in all the peoples. 99.9, right? Polypeptide, they are same. Only 0.1% is different, and that makes the total different. Okay. Functions are unknown for over 50% of discovered genes. 50% discovered genes, we have no any idea about their functions, right? less than 2% of the genome code for protein. Means transcriptionally active. When we think about the transcriptionally active DNA, we understand that only 2% of the genome code for the protein. Repeated sequence make up very large portion of human genome. Repeated means these sequences are repeated many times. So they make up very large portion of the human genome. Now, seventh is repetitive sequence are stretched of DNA. Repeated sequence, they are the stretched of DNA sequence that are repeated many times, sometimes 100 to 1000 times repeated, right? They are thought to have no direct coding functions, but they shed light on chromosomes structure, dynamics and evolution. They shed light on chromosome structure, dynamics and evolution. They have no coding functions, right? Means what? They have no coding functions means they cannot code any messenger RNA, means they are not active. 8 means chromosome one has most gene, means maximum gene is present in chromosome first, and it is, what is the number of gene? It is 2968. 2968. And why it has the fewest gene? It contains only 231 genes, right? So largest chromosome is chromosome first, which contains 2968 genes, and the smallest one is our Y, which has fewest, means how much? It is 231 genes, right? Smallest gene means what? Okay, it is the chromosome, largest chromosome and smallest chromosome, not a gene, but it is a chromosome, right? Here it is given the largest gene is our dystrophin gene, and which one is the smallest gene? It is TDF, right? TDF gene. And this TDF gene, it contains only 14 base pairs. Right? Largest, smallest, then also largest chromosome and smallest chromosome. And ninth one is what? Scientists have identified about 1.4 million locations where single base DNA difference. Single base difference means single nucleotide polymorphism, we can say SNPs, pronounced as SNPs. SNPs, right? Single nucleotide polymorphism is there. This SNPs, how many? 1.4 million locations are there. Location means loci are there, right? Pronounced as SNPs occur in human. This information promises a revolutionize the process of finding chromosomal location for disease associated sequence and tracing the human history also. So these are the silent feature of human genome, right? Project, human genome project. First is this, how many base pairs are there? 3164.7. Second, average base present in a single gene, it is 3000. Another given here, largest gene is 2.4 million bases. Smallest gene means it has, I will tell you, it is TDF, it contains only 14 base pairs, right? Total number of genes is estimated 30,000. In very early, it is which is very less than 80,000 to 140 genes, right? And in this one, almost all, means 99.9 are same, identical in all humans, only 0.1 is different from person to person. Functions, when we consider function, third is here, 50% genes, means we have no idea about the what are the functions, like unknown functions are unknown for over 50% of discovered gene. Less than 2% gene, they are actively transcripted, right? We can say actively active for transcription. Repeated sequence make up a large portion of the DNA, right? Third one is, these are stretch of DNA which are not responsible for any coding, direct coding, right? But they are responsible for, means we can say here, they shed light on chromosome structure, DNA, dynamics and evolution, right? And this stretch, it is repeated many times, sometimes 100 to thousands also, right? Chromosome first, it is the largest one which contains 2968 genes, and chromosome, that is last one Y, it is the fewest, means one it has only 231 genes, and last one is scientists, they have identified 1.4 million locations, means where single base DNA difference is there, or we can say single nucleotide polymorphism is there, which is simply pronounced as SNPs, right? Okay, now in next class we will discuss about the application and future challenges and also human DNA fingerprint. Okay.